Today I'll talk about how to solve medical mysteries through machine learning. Every year, hundreds of patients face uncertainty when healthcare providers are unable to diagnose the cause of their symptoms. So our work, along with the entire UDN network, tries to provide answers for patients and families affected by these mysterious conditions. Here, I want to present you a case and show you how we solve some of those cases through a collaboration of genetics, bioinformatics, machine learning, and molecular biology. In this slide, we have a 14-year-old exhibited a severe developmental delay. He's wheelchair bounded, never able to walk. She can sit, and she has severe cerebellum atrophy and also a corpus callosum abnormality. As you can see in this brain MRI scan, the arrow points to the regions. When you compare to the normal brain, you can see a very clear difference in the cerebellum region, in the cerebellum region, and in the corpus callosum region. Well, this proband had an elder brother who died at age 28. And according to her parents, the brother had a very similar sequence of kind of a neurological decline. Unfortunately, we were not able to get the DNA samples for his brother, but there's enough reason to believe that disease is probably Mendelian, is due to genetics. So a whole exome sequencing was performed on the family, on the girl and the parents. As you can, for those of you who are not familiar with the whole exome sequencing, I have a kind of a small illustration at the bottom of the slides where you can see we sequenced primarily the coding region and the nearby kind of regions for this proband, for this girl. And when you compared her genome, her exome, to her parents' exome, you're going to find a new mutation, so-called the de novo mutations. You're going to find things that are different and things that are kind of combined, small insertion deletions, all sorts of variations. So in this particular case, the clinician was able to find rare variants in 13 genes. The question faced by the clinician is which one of those is a good candidate, or even one step further, which one of this is actually causal? To answer those questions, typically the clinical geneticist will need to investigate each of the single gene and try to narrow down the list. And based on the answer of those questions, they can either prioritize or eliminate some of the candidate genes. For example, one can ask, does any of the candidate genes, right, can known to cause a disease with matching phenotypes? The first gene listed in this table is ATP8B1. If you check this gene in OMIM, you're going to find that there is association, or that this gene can cause cholestasis. It's a liver disease. Given the extensive neurological phenotype or symptoms from our patient, it is hard to link this gene to be the causal gene for this patient. So it's probably has a lower priority and can be eliminated from the list. Of course, one cannot completely eliminate the possibility of a genome variant just based on a single line of evidence. But for simplicity and for presentation purposes, I'm saying that this gene can be eliminated from our long list and trying to further narrow it down. And sometimes it may not be so easy and clear cut. The second gene on the list is EPG5. In OMIM, it's known to cause something called a Vici syndrome. It is a neurological syndrome. It has actually overlapping phenotypes with this girl. It's a failure to develop a corpus callosum. And along with that, there are other phenotypes like cataracts and the hypopigmentation of the eyes, a lot of related to eyes. These are non-overlapping. So when you compare these, kind of can lower its probability. And based on the same principle, you can eliminate 2 more genes from this list. The next set of questions one can ask is the gene expressed in the brain, right? Given this is a neurological disorder. So one of the genes on the list is called OBSCN, the first gene in this table. If you look at expression in the GTAX database, which is a database profiles multiple tissues across human post-mortem human samples, right? This gene is clearly highly expressed in muscle and heart. But when you look at the brain, it's expressed at much lower level. Of course, as I emphasized earlier, a single line of evidence is not enough to eliminate it, right? But for kind of in this presentation and for simplicity, we can actually reduce this the priority of this gene or the probability of this gene and kind of remove it out of the list. And we can further narrow it down. Another typical question that one can ask is about the particular variant. The first two was kind of focused on the gene itself, right? Is the gene expressed? But ultimately, it has to be down to the variant, the particular variant carried out in the patient's genome. So when we ask the variant, we ask a question like, can we, in a healthy, so-called healthy control databases, right, like NOMAD, can we find other samples, other people, right, that carries the homozygous mutation of the same variant, homozygous variants, the same variants, in other words. So oftentimes, if you observe a homozygous in a healthy control, and you observe a large number of homozygous in a healthy control population, there's a good chance that it is not causal. It's not actually damaging or causing severe phenotypes like this one. And based on that principle, the first gene AP2A2, we will observe 4 individuals, right, in the NOMAD database that carry the exact same variants, but in a homozygous form. So similarly, you can look at these variants and ask what is the observed frequency in the healthy control population versus the theoretically expected frequency. If these 2 numbers are very close, you will have a value of 1. That's the column O/E. And that will give you a loss of a kind of a tolerance to loss of a function, and you can kind of eliminate or reduce their probability. On top of that, in addition to checking the human variant population database, you can also run computational algorithms, which based on sequence conservation across evolution, right? By comparing human genome to apes, to other mammals, to flies, or even down lower on the evolutionary ladder, right? Can you actually find high reservation, right? Or protein function is in the domain of a specific protein domain, right? Is a mutation in that domain kind of impact the function? There are algorithms that can produce a score, quite a number of them. In this slide, I listed a part of it, and like CADD-FRAD, which is the first one, right? It spit out a score, an arbitrary score actually. In this case, it has a score of 21. So some other tools like Polyphon2 at the bottom will spit out a categorical variable. In this case, it will say it's a benign in contrast to damaging, right? So if you normalize it into a 0 to 1 range, with 0 being low probability of it's damaging and 1 being high probability is damaging, you can kind of average across all these prediction tools and trying to get a consensus. In this case, we were able to kind of run these DBNSF, which is a consensus database that averaged a lot of, combined a lot of these tools and provide a summary value. Here is 28.3% of chance that this is likely damaging, which is pretty low. So based on these tools, you can further remove variants in your list. And then we end up with 3 variants. where do we go from here, right? The clinician, you have exhausted the literature, exhausted the various databases. How do we move from here? Well, so what happens here is the clinician faces a question. I was able to narrow the entire 13 genes into 3, right, and their associated variants. How do we move further? We can actually bring in the model organism researchers or basic science researchers here, because some of them may happen to be working on this gene, which overlaps with the clinician's shortlist. And maybe the phenotype matches, the mechanism matches. The question asked by the basic science researchers is does my gene have a disease relevance, right? By combining the 2 sources together, sometimes we will be able to solve these cases. Here, I'll present you a successful case that's published by a talented postdoc, Dr. Yang at Hugo Balance Lab. In this publication, he was able to find out of the 3 genes that we talk about, the OGDH1 is actually the one that caused the patient's symptoms. And to do that, he actually harvested or used the power of a fly genetics. He was able to express the human gene in the fly, and also the human variants, the variants carried about the patient in the fly. And he was able to show, using rescue assays, he was able to show that when you express the human gene, you can rescue the fly. But when you express on the mutant form of the human gene, you fail to rescue the neurological and the lethality phenotype here. This is only a small piece of experimental molecular evidence or genetics evidence from that paper. There are more extensive studies, but by combining the works in the fly, in a model organism and the human genetics, right, they were able to solve this case, ultimately solve this case. So here I've presented you an example as how one can solve, or how a UDN case, undiagnosed disease network case, can be solved by using human genetics, bioinformatics, and the molecular biology and model organisms. If we generalize this a little bit more, right, on the clinician side, if you have n equals to 1, these rare diseases, right, you found a few hundred genes. Sometimes you're not lucky to narrow it down to 13. A typical input may be 100 or a few hundred genes. You have 2 questions. One is, how do you create a short list? Another one is, the second one is, based on that short list, how can we find the causal genes? I've shown you that involves actually to create that short list that involves queries on various databases, right? OMIM, HGMD, Decipher, all these human databases, right? And CAD algorithms that predict the pathogenicity, benign or damaging on the protein function, literature search of interactions, publications, expression patterns, and also various model organisms data mining. To streamline this, because if you're going to search 20 databases per gene, that work is not going to scale up. It's going to kind of slow you down rather quickly. So realizing this bottleneck in the UDN network, we teamed up with Claire Hu, a talented analyst at Paramount's lab at Harvard, and we created a search engine and a database that integrates 3 resources together: the human genetic resources, such as those Nomad and CleanWar, and the model organism resources, Flybase, MGI, Yeast, and trying to connect them through a homolog mapping tool called DIOPT, developed by Claire. And to put it even more simply, you can think of MARRVEL as the Expedia for rare disease research, where you can combine and streamline all the information, the data, relevant data, into a harmonized and easy to view a presentation visualization. There are other tools in this field, like Monarch and Ensemble VEP or VARSOM, right? Compared to those tools, some of those tools are not visually available, like VEP. It's more like an annotation tool. It's more of API based. MARRVEL is much more interactive. It's much more visual. It's online, and it actually covers a lot more resources related to rare disease diagnosis. So this project basically is a data wrangling process to kind of harmonize the data, put all the resources together. Since the start of this project, we have acquired a decent number of users, about 47,000 users and 300,000 page views across the world. We're pretty happy about the users, and we're trying to figure out what's next, right? Kind of to give you a little bit more impact of MARRVEL on the UDN project, right? Through extensive model organism studies and the use of MARRVEL, we were able to provide a lot of new scientific insights during the phase 1 of the UDN. In the phase 1 UDN, about 2,000 applicants applied. We were able to select half of them, about 900 cases. And most of them were sequenced, their parents or sibling was sequenced, and about 200 variants were submitted, a total of 122 cases. Through MARRVEL, we were able to further narrow it down to 73 genes. 59 of those genes were studied in the fly model organism, and 16 of those was studied in the zebrafish. 2 genes were studied in both, actually. And the table here presented the findings from the phase 1 or from the fly core and zebra core. So all the black ones, the genes that in black color, are those that are represent phenotypic expansion, meaning that observed clinical features extend beyond those reported in the associated known disease gene databases like OMIM. In other words, we were able to link additional new phenotypes that are missed by the original OMIM database or HGMD database. All the red genes that are in bold font are about 19 of them. Those are the genes, the novel disease genes that led to direct diagnosis by using a mask data, the fly model organism data. And those are red, but not in the bold fonts, are the potential gene discoveries. Well, what's next? How can we do further? We finished the data wrangling part. Can we do more, right? Well, the question as a data scientist and bioinformatician is, can we develop a machine learning system, right, that can assist this process, can maybe do as good as the human, or maybe even better, right? Can we do that? There's a lot of talk about artificial intelligence, right? Nowadays, we see that actually in our daily life, from recommender systems in Amazon, right, to search engines, to our face recognition apps. Really, there's a lot of machine learning or AI systems in our life. Of those, oftentimes you require a large amount of training data. But what we are meaning that you need to teach the machines again, right? The challenge we face here is we don't have a lot of these cases, especially for rare or undiagnosed diseases. The number of cases that we can use to teach the machine is quite limited. So how do we do? How do we do this problem? How do we tackle this problem? Here is kind of an overall schema of our approach. The input for the AI system, for the machine learning system, is the personal genome and the EHR, electronic health record, phenotypes of the patient. We can use MARRVEL to convert this into more computable data, right? For example, the EHR data sometimes is is natural language in the form of natural language. How do we convert it into more computable languages like the HP human phenotype ontology, right? And a lot of these data wrangling was handled by MARRVEL. But when it comes to the learning part, there are generally two categories of approaches. One is the black box approach, like a lot of these deep neural networks, where they have very good performances in recognizing a phase, recognizing a particular problem. However, these oftentimes require, as I said, many trainings, right? There are researchers kind of do one shot or several shot learning that reduce, but still they need to pre-train. And another approach is more like a hybrid knowledge-based system where we can integrate our biological, in this case is the human genetics knowledge, into the process and trying to derive a system that is more interpretable. We decided to go for this hybrid model instead of a kind of deep learning model. Just to give you an idea, an overview of the data we have, for the training, to train this model, we have about 495 samples from Baylor Genetics, the department. And out of those, 400 were used for training, 99 were used for kind of a validation. The training part was used to find the proper tuning parameter. In this case, we used a random forest. And in other words, we need to use this training to figure out what's the tree depth, the number of trees, right, and so on and so forth. And we reserved 126 samples from Baylor Genetics as a testing, and then we used 88 diagnosed case from UDN as the testing. So on average, if you look at the lower left corner, the graph is number of variants present per sample is in the order of 10,000. This is without any filtering. This is just kind of the basic input. After initial filtering, we're dealing with about 1,000 variants, 1,000 to 2,000 variants per sample. And if you look at the diagnosis, right, which is this pie diagram at the bottom, about 80% of the cases was solved by one variant. In other words, it's a single gene disease. About 20% of those are solved by multiple variants, and it's a multi-gene disease. So that's kind of the basic summary statistic. To approach this, right, as we proposed earlier, we're going to do a hybrid model. In other words, we take these raw features, the variants, the scores, we come up with 6 modules by asking questions, relevant biological questions, such as the one I showed in slide number, in the first few slides. Is the variant, can the variant cause a known disease, and it overlaps with the phenotype of the patient, right? It's how each decision is made and how we reach the final score of low, high, and medium. But to give you the sense of complexity, these type of questions involves a lot of manual curation about the decision modules and the questions that we ask. And we try, in other words, we're trying to find a function to code these biological questions into computable features, right? This may or may not work. That's our hypothesis that it can improve, but without running and testing this algorithm, we don't know. Once we did the feature engineering, the next step is what kind of machine learning algorithm we're going to use. We tried a couple. Finally we ended up with a random forest because it's kind of interpretable and also quite powerful in terms of the accuracy. And by running these, for those of you who are not familiar with random forests, think of it, it's a multiple decision trees, right? For each tree, you're going to answer a yes/no question, and it will lead you to different decision paths. At the end of this tree, you will reach a conclusion. Hey, this is causal, this is not causal. If you have 100 trees, at the end, you'll do a majority vote. If maybe sometimes 50 of the trees voted causal, the other 50 voted no, right? Or maybe just one tree voted causal, the 99 trees voted no, right? Based on that's your kind of final output score, how many trees voted causal, and then you can rank based on that score. Presumably the variant that ranked in the top one should be more probable, more likely to be the causal variants. And ideally, we want to rank this into the top 5, our causal variants into top 5, because think about the short list that you have, right? The model organism people will, to investigate each of the variant and the gene probably take 6 months to a year. If you for every case, if you give them 3, that's already a lot. The first one works, right? Otherwise, the second one, the third one is going to take years to figure it out. That's a lot of time and resources. So we kind of benchmarked the result to another algorithm in the field, Axomizer, and we were able to see a promising kind of competitive edge compared to Axomizer. Again, I have to kind of throw a little bit grain of sauce here that this is all our work is just recently done, so we didn't really optimize it much. And for the comparison Axomizer, we have 2 students working on it and running it, get similar result. It's not completely optimized, so in order to kind of publish this work and really get a fair head-to-head comparison, maybe both methods needs to be further optimized, but this is kind of running by default parameters, and this is what we got, right? If you look at what is the chance that a variant will be, the causal variant will be ranked into top 1, using MARRVEL AI, 68% of the chance we were able to do that. And in Axomizer, it's about 47% of the chance you can rank the causal variants in top 1. If you think about the top 2, in other words, what is the chance that the causal variants will be ranked in the number 1 or number 2? And we have about 74% of chance, and Axomizer has about 63% of chance. On the UDN cases, it's kind of the trend is similar. The numbers differ a little bit, not too much. 60% versus 36%, 71% versus 57%. The next few questions we want to kind of ask: did our feature engineering work, right? That's our initial hypothesis, but how do we figure that out? First, we need to find out which feature is more important. To do that, in random forest, there's something called a Gini index based on the entropy, the information entropy, to kind of figure out which feature is more important. We ranked based on the Gini index, and the top 6 features is listed here. First one is the diffused Frank score using string. And Frank score is actually a tool developed at Stanford that measures the phenotype similarity. And we actually did something extra on top of it. Based on those scores, we did a diffusion because there are many missing values. If you only directly apply Frank, some of the genes are not in the original Frank database, right? And the Frank doesn't have a Frank score. So we use the protein-protein interaction network and then diffuse it over. And in other words, we did an imputation. Some of them that are related, some of the top features are related to OMIM and the nomad allele frequency and the tier AD. That's our kind of inheritance mode feature. Well, we know which feature is important. The next step is if we remove those important features, how much that impact us, right? So we did that, and if you can see here that when we remove the first feature, the diffusion Frank, the performance didn't drop much. The first column is the one using all the features. The second bar is the one trained and tested by removing the diffusion Frank string feature, the number 1 feature. This indicates probably there are a lot of feature redundancy. We removed the top 6 features one by one. The performance didn't really drop much. indicating there's quite a number of feature redundancy here. What if we remove all of the 6 features altogether? In this case, it did drop from 92% to 83%. What if we just do not use any of the engineered feature, only use the raw features? In this case, it dropped quite a bit to 72%. In other words, the feature engineering work that we did indeed improved. But it's difficult to say which one because there's a lot of robustness among these questions, right? Multiple lines of evidence, they are complementary to each other. So it's difficult to say that if I remove a few, it's going to kind of deteriorate, the performance will deteriorate. So the next question is, can we figure out the decision trees, whether some of the decision trees can learn a real biological question, because the trees are constructed by an algorithm. Does that represent any real biological questions? One of the questions we have is, can it learn the inheritance mode, right? The dominant versus recessive. Well, some of the tree happens to put these together, right? And when we looked into our trees, we were able to find one of the trees indeed put this together. It has 2 decision node here in this dashed red box, right? One is AR, tier AR indicates is autosomal recessive. Another is AD, is actually autosomal dominant. When we run an example through this pipeline, if you look at the bottom of the slide, which is decision flow, so this gene is not curated in OMIM. The similar phenotype was found in HGMD. It is not likely to be autosomal recessive, but it is autosomal dominant. So these 2 decision nodes put together indeed kind of confirm the inheritance mode. Next, we want to further understand how the random forest works, and does it work differently on 2 types of cases. The first type, we divided the entire data sets into those genes, those known disease genes and disease-causing genes, known diseases curated by databases such OMIM, HGMD, and the second half is those that have unknown disease genes, and basically the symptom is totally unknown. So for the first one, I'm going to present your case and trying to explain how the random forest was able to mimic the human experts and then reach this very good conclusion, was able to prioritize or predict the causal variance in the number 1, in the top 1. And in this patient, this family has 4 boys. One of the boys is affected, and this boy has a lot of neurological features like seizure, delayed speech, and also some abnormalities of the face and dentures. And if you look at our prediction result, which is on the left, and each dot here is a variant, and the y-axis is the prediction score. The higher the prediction score, the more likely it is a causal variance, right? It's prioritized higher. So if you look at the ranking, we sorted all the variants based on this prediction score, right? The number 1 is a gene called ATP183, and it's red because it's actually, if we look at the genetic diagnosis provided by the clinician, this is indeed the causal variance. And how did we reach the conclusion? If you look at the decision module, at the bottom, the first decision node is whether the gene is curated in OMIM. In other words, it's checking, right, that is this a known disease gene and disease-causing gene. And then second one is checking what is the allele frequency, because we're dealing with rare diseases, and if a disease if a variance has a very high frequency in the healthy population, there's a very small chance that's going to cause a rare disease, right? And then the next few questions is about the phenotype matching and whether the ClinVar is about the pathogenicity scores, right? So if you look at the decision module put together, it's very logical. It kind of bears the same logic as the clinician uses. And if you look at the phenotypes from these, on the left side is the patient phenotype. On the right side is the phenotype from the OMIM, curated from OMIM. And as you can see, there are a lot of overlap: seizure, global developmental delay, dystonia, right? The weakness of muscle, and also the conical tooth, the denture issues, and abnormality of the face, right? So this case is considered solved based on the 3 lines of evidence: OMIM curation, frequency of allele, and then the molecular evidence on this on the gene and the allele. So in the next group, we're trying to see, well, the random forest was able to learn and diagnose a known disease. What about known but rare diseases? But what about these unknown cases? How can they solve it? And presented in this one is a family that has single child, a single girl, and it's affected. It has a lot of a global development delay like the previous one, epilepsy, seizure, and other things. If you look at this case is solved, and the causal gene, if you look at the causal gene predicted by random forest, the number 1, right? The green node says the gene is not curated in OMIM or HGMD. So in other words, this gene has no known association or causal relationship to other diseases. So how did we rescue this, right? It's new novel gene variants and disease-causing gene by looking at the other evidence. In this case, a GERP, for example, it has a high GERP score. The GERP score is about the evolutionary conservation, so the mutation is in a highly conserved region, and it has also high polyphen scores. That's also conservation. If you look at the PLI score, that's from the NOMAD, and that tells you what's the tolerance of loss of function. It's not really tolerant for loss of function, right? And that's kind of how we reach the conclusion. And VEP is also high, so it's a low frequency. Combining, this is only 2 example trees. About 30% of the trees actually raised this is a causal using a lot of complementary informations. Well, this is a case of n equal to 1. How was it ultimately solved? Because the computational work we gave you can only prioritize. It doesn't really solve it, right? So if you look at the publication on this one, it is solved because once you've known this case, you're confident about this variant, you can go to other databases like GeneMatchers, MyGenes, other kind of consortium rare disease databases to see if there are other cases like this that happens to carry same or similar mutation. In this case, our case is number 9. And as you can see, there are 3 other patients that has the same mutation in the same location. It's an early termination mutation. And there are other 7, 8, and 9, right? 3 more cases. 2 more cases, 10 and 11, that has the same early termination mutation, but it's in approximate regions, that 5 base pairs after our patient. So really, this is a mutation hotspot. There are multiple patients have similar phenotypes and carry the same mutations. And it is rare, but once you're confident about this mutation, you go out and search other databases, there are probably more patients like this that are undiscovered. And that is another second challenge, actually. How do you kind of go beyond a single silo of disease cohort and kind of bring the power of multiple families and multiple consortium together to solve these, right? Of course, the group also went into the molecular mechanism and realized that the early termination of this gene can cause a diffused localization in the nuclear body that I'm not going to dive deep into, but it not only they were able to observe multiple patients, but they were able to find the molecular mechanism. Hopefully that will lead to a discovery, drug discovery. Well, so far my talk have focused on kind of bringing the two community together, the clinicians working on the patients and the model organism, basic researchers working on the genes. Combining these together, we've shown again and again that we can actually facilitate this. But there's more need to be done actually, because these communities don't really work together or closely work together enough. We want to kind of push the overlap. There are existing databases like a GeneMatcher, right, Matchmaker Exchange that can help you query genes. If you have a variant, you want to find are there any patients have the same variant and similar phenotype, you can submit that. There are multiple rare disease patient family registries and network communities, and there are scientific basic science communities that are working on individual genes. We really want to create a common base. There are some existing forces like the one from Canada, RDMM. It's a funding-based human model organism matchmaking process. In other words, a clinician can nominate a variant and a gene, and then the Canadian RDMM will match their interest to a basic science, maybe scientists maybe studying C. elegans, yeast, or fly, and a certain amount of money will be given to this project to kind of invest this variants and gene once the matchmaking is done. And similarly, there's one in Australia, European, and Japan. And ModelMatcher here is using the same APIs. Actually, we have very close collaboration with the RDMM group at Canada, and we're developing a platform that can really talk to this diverse community, the rare disease consortium communities, the basic scientific research community, trying to bring them together. So I'm not going to go into detail how computationally this is done, but this involves a lot of a collaboration with our collaborators in Canada, and I'll show you kind of an example how this is done, right? The basic scientist research can nominate a few genes, A, B, C, D, E, F, and there are certain level of privacy. You want it to be public or you want to be restricted. And for clinicians, no registration is needed if they only want to search a public profile, but if they want to search the restricted genes, they need to register and do the match, right? And for basic scientific researchers, the genes can also be prioritized into, oh, this gene is my priority, I really studied it. Or this is a secondary, right? A gene where I studied it maybe many years ago, the lab member has left, it's inactive. So for clinicians, so when they search it based on this priority, we'll kind of do this match, trying to prioritize and match a proper team. And so far, we have finished the phase 1, where we have launched the scientific registry, the ModelMatcher.org. People who are interested, basic scientific researchers who are interested in participating in this can register. And we are working on the phase 2, which is the clinical registry, trying to recruit clinicians to register. And the third phase will be to able to talk to a matchmaker and GeneMatcher, right, to kind of link to other public domain databases. And here is our ModelMatcher.net website. Please follow us, register if you're interested. And just to give you a little bit of statistics here, in total, we have about 500 ModelMatcher users. RDMM from Canada has 190, and European has 189. But this is the public profile. Some of the profiles are private. When we talk to the Canadian collaborator, they have about 700 users additional users that are private. They didn't make their profile public. So really, there are about like close to 1,000 users, model researchers registered. We assume the same number is true for the European. So we just launched for a couple months. So we are aiming to increase our register from 100 to maybe to 2,000. And this is kind of a layout of the type of model organism researchers, right? People studying mouse, fly, frog, yeast, zebrafish. There's quite a diverse groups here. And with that, I would like to end my research by acknowledging many of the collaborators involved in this work. It's really a team science. Shinya Yamamoto and Michael Harnish is the kind of conceptualized the ModelMatcher, and our lab is the kind of informatics lab. Lucien and Songyang developed the ModelMatcher platform, and our Canadian team, RDMM team, Phil Heiter and Kim Boycott really is the provided the original source code and a lot of valuable informations. And the MARRVEL AI team involves my lab. Dongxue is a very talented postdoc who kind of led the entire project along with Rami. We have 3 students work on the machine learning part, Lin Hua, Chao Zhong, and Ko, and our talented programmer, Songyang and Lucien, provided a lot of programming support. And most importantly, our collaborator on this really taught us how geneticists diagnose their cases. Without them, we will not be able to do this feature engineering. Without them, we will not able to actually do any of the work on the MARRVEL AI. That's Peng Fei, Bo Yuan, Hugo Ballon, Brandon Lee, Shinya, Michael Wrangler, Julia. They all kind of contributed significantly to the success of our work. And also the funding agencies from Child Foundation, NIH, Texas Children's, UDN, and our family foundation from Child Family and the Huntington Family. All right, with that, I would like to end my presentation. If you have any questions, you can ask me.
Okay, now you can unmute yourself to ask question or type the question in the chat. Let me check the chat. Okay. So this one question, what is the main challenge in developing the AI system for rare and undiagnosed disease?
Yeah, well, that's we spend a lot of time doing data wrangling, actually. The tremendous amount of time was trying to understand what is the output of a data column. Because sometimes we have tons of missing values, and we're trying to figure out why, how can we impute them, because most of the existing machine learning algorithms are not friendly to missing values. And there's a lot of categorical values, and we're trying to make them more quantitative. We're trying, that's where we spend a lot of time really to understand our data. We didn't really spend too much time on selecting the exact algorithm compared to the time we spend on data wrangling, actually. So we probably will still explore more on the model, the actual machine learning model going to be used. Oops, can't see my mouse here. There is a question from Anita. How much cleanup did you need to do on the CleanWAR database? Yes, that's a great question, actually. As a data scientist, I don't understand the CleanWAR at all. I learned it from our collaborator, in this case, particularly from Peng Fei and Dongxue and several other clinical geneticists. It's a lot. That's why we did a feature engineering. There are a lot of submissions, right, that are noisy, and sometimes a single submission is actually valuable compared to tons of submission, right? There are benign, there are conflicting evidence, right? Some submission will say this is benign, some submission will say this is pathogenic, likely pathogenic, right? So yes, we did combine these into a more engineered features rather than just rely on it. For example, how many submissions are there, right? If you are giving a pathogenic score, how many are pathogenic? How many are likely pathogenic? Even like what is the group? Which group did submitted this? Is this a large clinical group or this is like a one source?
Yeah, so it's from Ron. I think it's a comment. So said dkNET may have much to offer through the Hypothesis Center and the Signal Pathway Project, SPP. The focus of SPP is to make high confidence connections between genomic targets and their upstream regulatory pathways and disease states. So this could help for further define both function and network connection. Yeah.
Absolutely. I mean, we are actively expanding the feature space, right? For machine learning to work, I think a lot of the useful features or strong features is indeed needed. Like model organism data is still not into this machine learning part yet. And I certainly think more pathway related information will be useful.
Mm-hmm. So next question is from Nick. So great work. Are you training ingesting structured EHR data only for your random forest?
Yes, so far we are only doing the structured EHR. Actually, we converted into HPO, the human phenotype ontology G data. Actually, a lot of these, some of these were annotated by Baylor Genetics, a UDN network. We also have another tool developed by Cole Dysarov, one of the students, where he developed a tool that can convert a natural language into HPO, CleanFin, and that's kind of how we compute on natural language or EHR data.
Okay. So next question is from Chen. Do you consider updating the model with more data coming in? If so, how often do you plan to do that?
Right. So I think a lot of data are existing in silos, right? There's the DDD data in Europe. In Europe, there are multiple other data sets. So that's really requires a lot of efforts, and that's a frustration, I think, for many people working on human data because of these barriers, IRBs and protocols, right? Like it's difficult to get access to a lot of these data. And we are part of UDN, so we have access, and we are part of Baylor, so we have access to Baylor. So that's kind of even for that, it took us a lot of time to get access and to clean up the data, especially the human data. So yes, with more data, it's going to be more powerful. We certainly agree with that. At this point, I think we need more people actively working on the data wrangling part. It took us a lot of time to wrangle the data.
Okay, thank you. So next question is from Kristen. How did the group decide which model organism to use for biological testing? In the cases we saw flies as versus zebrafish.
I think this is a question I probably don't have the right answer. Of course, it's can be. I don't have an in-depth answer. I can give you partial answer. It's sometimes it's through ortholog, right? And it depends on the phenotypes and the availability of the reagents, right? And the phenotypes. But if you look at it, the fly core did a lot more than the zebrafish core. I didn't involve in the decision of whether it will be studied, or like which variant will be studied in which model organism. Let me see if one of our team member is here, so he can give you a better answer than I. But it's through kind of ortholog matching and phenotype matching of reagents, availability of reagents. If you want an in-depth answer, I can connect you to Michael Engler and Schema, who kind of handle this part.
Okay, so next question from Ivan. How does MARRVEL AI map natural language from clinical notes into HPO terms or other
Right.
structured data?
Right. The two approaches. One is manually edited, right, from provided by Baylor Genetics and UDN. Another is by CleanFin, which is a program that can automatically convert clean notes to HPO.
Okay, and then there's one more question. Can you elaborate on the added benefit of using whole genome sequencing compared to whole exome sequencing in disease diagnosis?
Sure. So about 30–40% of the case that go through Baylor Genetics can be diagnosed. The remaining 70–60% of cases are not diagnosed. So can we increase the diagnosed rate? People are moving toward whole genome exome. In other words, looking at the regulatory regions instead of the coding regions. We are also kind of combining RNA-seq technology, trying to look at other splicing variants. So by looking at more places, we hope that we can find causal variants and give us more power and more resolution, right, on each individual case. However, there are a lot of challenges in the whole exome, whole genome sequencing, because you are increasing the search space by a lot, right? So that's, and oftentimes it's much harder to validate the regulatory regions, especially using model organisms, right? So there's quite a number of new challenges for using the whole genome sequencing data, both computationally and at the experimental validation level.